This video will focus on proteins and their synthesis. So we'll discuss the genetic code, basic aspects of protein structure, and the events involved in translation. Let's begin with some classic experiments done by Beadle and Tatum in the 1940s. They worked with a very common mold called Neurospora, and based on their observations, they came up with the so-called one gene, one enzyme hypothesis. What they did is working with this mold that involved a very basic metabolic pathway shown toward the bottom of the screen. So this mold requires, on the far right here, the amino acid arginine. It can either take it up from its environment, or it can also synthesize arginine on its own. And it can synthesize arginine by the pathway, whereby it takes some precursor molecule that then gets converted to ornithine, which is then converted to citrulline, which is then converted to arginine. And notice that each step along the way requires a specific enzyme. And so what they did was they took some mold, exposed it to ultraviolet light, which induced mutations. And it turns out those mutations were in each of the three enzymes shown in this metabolic pathway. So they then took mold that was unable to synthesize arginine, again due to the mutated genes that code for either of the three enzymes, and then they grew those strains with the following supplements, either with ornithine, citrulline, or ultimately with arginine. And so think about that setup, and then they simply assayed to see which strains were able to grow under these various conditions. So here's the setup and their results. So some strains, of course, where they didn't add any nutritional supplement, they could not grow. Or on the far right, if they added arginine, the end product, of course, they could grow just fine. And this first strain that they isolated, this, it turns out, was a strain where their mutation was in a gene that codes for the very first enzyme in the pathway. So just to think about it, if you notice the pathway toward the bottom, if this first gene is mutated, that codes for enzyme 1, if you add ornithine, you would expect it to grow because it can complete the rest of the pathway. If you add citrulline, again, you would expect it to grow. And of course, if you add arginine, you should expect it to grow as well. And so short enough with that strain, that's the result. Those are the results that they ended up with. Another strain, again, that was mutated, the gene that codes for the enzyme 2 was mutated. And so again, think about the pathway. If you added ornithine, that would not allow the fungus to grow simply because you don't have the second enzyme here that can convert ornithine to citrulline. However, if you had citrulline or arginine, again, you would expect those to grow. And sure enough, that's what they found in the second strain. The third strain, again, same logic. That strain had a gene mutated that code for the third enzyme. So ultimately, the takeaway message here is based on their results. Again, they came up with this one gene, one enzyme hypothesis. In other words, genes code for enzymes. Now, of course, we have expanded that, and we recognize that genes code for all proteins, not just enzymes. So now let's think more broadly about information flow and think about the central dogma of molecular biology. So you've seen this before, this is review, but imagine DNA transcribing a piece of RNA, which then leaves the nucleus, enters the cytosol, and then it can be translated to form some protein. That's what we're going to take a look at now. So imagine a piece of messenger RNA that's just been transcribed. Now essentially what happens to that messenger RNA once it's been processed and so forth? Now it's a mature messenger RNA. What happens next? Well, again, it leaves the nucleus, it's translated into a polypeptide, which is eventually processed into a functional protein. So let's begin with a review of the genetic code. What we see here is the genetic code table. Recognize that codons are typically in a 5' to 3' direction. So codons are, of course, the three bases found in messenger RNA that code for specific amino acids. And you see that illustrated here in this table. And imagine if you have some DNA sequence shown toward the top here. Again, directionality is indicated, so 3' toward the left, 5' toward the right in this particular example. In order to figure out the sequence of amino acids that would be found in the protein if this gene were expressed, the first thing you simply do is transcribe that to a piece of RNA. That's what we see here. Identify your start codon, and your stop codon, and then essentially use the genetic code to figure out the sequence of amino acids based on the codons between the start and stop codons. Notice a few things about the genetic code. First of all, it's redundant, or in other words, degenerate. In other words, what that refers to is the fact that for many of these amino acids, take a look at serine or proline, there are multiple codons that code for each amino acid. Notice also that the code is specific. In other words, every codon codes for a specific amino acid. I should also point out that it is evolutionarily conserved, meaning that the genetic code that we see here holds true for humans, for dogs, for fish, and even bacteria. So the same genetic code is used in all organisms. A quick review of protein structure. Recall that there are various levels of organization that we can find in proteins. So we can refer to the primary structure of protein, which is simply the sequence of amino acids. Secondary structure refers to these commonly occurring structures, namely a helical shape like we see here, the alpha helix, and a pleated sheet structure like we see here. The tertiary structure of proteins, that just refers to the overall three-dimensional shape of a protein. And those proteins that consist of two or more chains of amino acids or two or more subunits, we say those proteins display quaternary level of organization. It turns out that amino acids are linked together by condensation reactions or dehydration synthesis. This, of course, occurs in ribosomes. We'll come back to this in just a few moments, but I just want to point out here that here we see three amino acids being linked together, essentially forming peptide bonds, which are just covalent bonds between those amino acids. And notice that every time that occurs, you remove a water molecule. So that's what's meant by a condensation or dehydration reaction. Now let's think about the process of translation. Here are some of the key players. First of all, we need a piece of messenger RNA that was produced by transcription. We need some tRNA as well. tRNAs, these are RNAs that ultimately can include an amino acid on one end of the RNA molecule, and they also include an anticodon, which is essentially three bases that are complementary to codons. So this is illustrated here on the far left. So here we see a tRNA molecule, 3' end, that's where an amino acid will eventually be attached to. The anticodon shown down toward the bottom here, again, complementary to a specific codon. And as of right now, this particular image, there's no amino acid attached to the 3' end. But if that tRNA has an amino acid ultimately attached to it, it turns out there's an enzyme, Aminoacyl-tRNA synthetase, that charges the tRNA. In other words, attaches an amino acid to the tRNA. So here we see the enzyme shown in blue attached to a piece of tRNA and also attached to alanine. So essentially what the enzyme does is takes the alanine and again links it to the 3' end of the tRNA. And it's illustrated to the far left. Here's a tRNA that happens to have serine attached to the 3' end. So again, this one has been charged. So in addition to mRNA, tRNA, we also require ribosomal RNA. Turns out ribosomal RNA is part of ribosomes. Ribosomes are fairly complex. They consist of lots of different proteins as well as ribosomal RNA. Here's a figure that illustrates ribosome structure in both prokaryotes and eukaryotes. Again, notice that they can be fairly complex. They can include various types and sizes of ribosomal RNAs, as well as a lot of different proteins. But I just want to point out that whether we're talking about prokaryotes or eukaryotes, ribosomes consist of a large subunit and a small subunit. Okay. And often in the cell, those two subunits exist separately, but during translation, they come together and function as a single ribosome. So how does it all begin? How does a ribosome know where and when to initiate translation? So in prokaryotes, there is a specific sequence called the Shine-Dalgarno sequence, illustrated here. Notice this sequence is just upstream of the start codon. So essentially what happens is there's a sequence that's recognized the Shine-Dalgarno sequence is recognized by this 16S ribosomal RNA. It turns out, I'm going to go back real quick, the 16S ribosomal RNA, notice that's part of the small ribosomal subunit in prokaryotes. So essentially what happens in prokaryotes, the small subunit recognizes this sequence, it binds there, and then the large subunit comes in, and then things are ready to proceed during translation. That's also illustrated here. Again, here's a piece of mRNA, small subunit binding to the mRNA. The first thing that happens is a tRNA that recognizes the start codon, enters into the system, and then the large subunit joins in, and now we have a fully functional ribosome that can undergo translation. And so in eukaryotes, initiation, similar idea, but just slightly different. We don't have a Shine-Dalgarno sequence, but the small subunit recognizes the 5' cap. It then slides to the start codon. That first tRNA carrying methionine is added, and then the large subunit comes in as well. If we look at the structure of ribosomes, there are various sites or regions that we identify that play various roles, namely the A, P, and E sites. So if we think about the A site, you might think about that as an acceptor or an aminoacyl-tRNA region. This is a region, of course, where the tRNA enters the ribosome that has been charged, so it's carrying its amino acid. The P site, you can think about that as peptide bond or peptidyl region. That's where covalent bonds are formed between amino acids. Then the E site, that's the exit site. That's where the tRNA is released after the amino acid has been added to the growing chain of amino acids that's exiting the ribosome. I will also point out that there is some polarity to a polypeptide or to a protein is produced. Noticed on one end, if you think back to the structure of amino acids, recall that on one end they have an amino group, and on the other end they have a carboxyl group. So we would say this growing polypeptide, this is the amino side, and then over here will be the carboxyl side. And notice how that correlates with polarity on the mRNA. So 5' end of the mRNA correlates to the amino end of a protein. The 3' end of the messenger RNA correlates to the carboxyl end of a protein. So elongation will take place. That's essentially the ribosome making its way in a 5' to 3' direction, reading the messenger RNA, bringing in appropriate tRNAs, whose anticodons are complementary to codons, linking up amino acids, which then, of course, exit the ribosome as translation proceeds. So initiation will continue to occur until a stop codon is reached. When ribosome encounters a stop codon, instead of bringing in tRNA with amino acid, there are release factors that come into play. And essentially, once the ribosome encounters a release factor, then the two subunits separate, the amino acid chain is released, and eventually that will be processed into a fully functional, mature protein. So that concludes our discussion of proteins and their synthesis.